Hi, everyone, and thanks for coming out. My name is Wasan, and I'm going to be talking with you today about my NMIN project, the Lipidic nanoparticle formulation of a triple adjuvant for intranasal mucosal vaccines. I work on this project with co-PIs, Dr. Volker and Yanzu, and co-investigators at UBC, Dr. Bob Hancock and Dr. Peter Wasan. What I'd like to talk to you about today is our lipidic nanoparticle formulation of a special adjuvant that's been developed at Vito, looking at intranasal vaccines. And this project is focused on pertussis and influenza, but for the purposes of today's talk, I'll mostly be talking about pertussis, which is also known as whooping cough. So we'll talk about why we still need improved vaccines for pertussis. I'll introduce what is a mucosal vaccine, what are their advantages and limitations, and how this might be beneficial for pertussis and other vaccines for preventable respiratory illness. We'll learn what is triage, this combination triple adjuvant, and the approach that I've taken to develop a carrier for triage to facilitate intranasal delivery as a mucosal vaccine. We'll introduce the physical features of L-triage, the lipid carrier for triage, and different fabrication approaches that I've been using. And we'll look at some immunogenicity studies with both model antigens as well as pertussis, and talk about the challenges and ongoing development plans. First of all, pertussis, which you may know as whooping cough, used to be a big cause of morbidity and mortality before wholesale vaccines were introduced, which really knocked down the incidence of pertussis quite a bit. So by mid-70s, it was very low level because people were vaccinated against it. However, by the 80s and 90s, people were sort of questioning the safety of the pertussis vaccine, side effects, and so on. And so in answer to that, an acellular pertussis vaccine was introduced, and the wholesale vaccine was pretty much phased out and is no longer available. However, going into the 2000s, we started seeing some epidemics, some outbreaks that raised concern. So it has been on the uptick, although not to the levels it was prior to the era of vaccination. So this is there's certainly room for improvement for the current acellular pertussis vaccines. So there are certain groups that are particularly at risk, and you may know the current series, DTaP, so diphtheria, tetanus, acellular pertussis, or Tdap, similar. It's a course of 5 vaccines. So it is not uncommon to miss a dose or two and not have the full series of 5. So in addition to that, and there has been some evidence of waning immunity over time, and we're still not quite having our best effect with the pertussis vaccines. So some people are particularly at risk. And even if someone who is unvaccinated gets sick with pertussis, it's known as a 100 days cough, and you can be sick for quite a while. We do still need vaccines that are appropriate for the very young, who are most at risk. With current parental vaccines, the maternal antibodies can interfere with that. We need to find ways of addressing the waning immunity that's seen, have an effective local immune response. It is a respiratory infection, so if we can reduce carrier status and affect a local response, that's your best bet. And it'd be great if we could have fewer doses as well. So can a mucosal vaccine achieve these goals? Might be ambitious to achieve all of them, but maybe we can make a dent. So what is a mucosal vaccine? So that means a vaccine which is applied to any mucosal surface. I'll be talking about intranasal vaccines, but it also refers to oral vaccines, like oral polio, or the urogenital tract, which is useful in agriculture, for example. But we'll be talking about the intranasal route. So as the vaccine particles or the pathogen enters into the nasal tract, as shown in the figure in the top right, it will encounter immune cells, for example, dendritic cells, which will take that material to nearby lymph nodes. It will track that antigen to lymph nodes, where it will encounter B cells and T cells to produce antigen-specific antibodies and activated T cells, and hopefully never be in T cells, which will then create a systemic response as well as a local response. So our vaccine nanoparticles that we're developing are intended to go by this route. So those dendritic cells have to kind of reach up and grab the antigen, grab the vaccine nanoparticles, and then bring it on to the systemic route for approaching the lymph nodes. So what we're trying to do is to generate both a mucosal and a systemic immune response. We want that secretory IgA, we want serum immunoglobulins specific for the antigen, mucosal effector cells, helper cells, cytotoxic T cells, and those memory B and T cells. So that's the endpoint that we're looking for. This local immune response is really important for respiratory infections in particular, through the intranasal route. And the further advantage with the intranasal route is it may actually reduce costs. There may be less ancillary supplies needed. We think about all the supplies and materials that are needed for parenteral administration. Potentially better patient compliance. There are people that are afraid of the needle, so it may be a little bit easier for them. And there is a potential to vaccinate the very young because of that lack of interference with circulating maternal antibodies. So basically, we have the potential through this route, and there's an increased interest in this route of administration. There's a lot of research going on with mucosal vaccines. We're focused in our lab on building these better vaccines for pertussis, influenza, and also 2 veterinary vaccines, bovine viral diarrhea virus, known as cattle shipping fever, and porcine respiratory and reproductive virus, which is important in the pork industry. Okay. However, this is a very challenging project. Think about the mucosal environment, right? So as we breathe in on a daily basis, we encounter many antigens, and we can't respond to all of them, or we would be just reacting all the time. So we're actually pretty tolerant of a lot of antigens that come into that environment. So we do need to use adjuvants to enable that response. In that environment, we also have mucus, all of its components, enzymes, the pH can vary just a little bit, physical barriers, and so on. The antigen has to be stable in that environment. Those adjuvants have to be appropriate in terms of toxicity and safety by that route. So we may need some kind of a delivery system. And of course, we have to do our, all our evaluations in that environment. There are limited in vitro models for investigating this route of administration. Those things are coming more into use, but the translation between an in vitro model and a whole animal is still a bit of a leap, and we need to have that correlation of the mucosal response to all the way to responding properly to challenge with the pathogen. Okay, so briefly, those barriers that we would encounter. We've got the mucus layer barrier, we've got clearance mechanisms in that environment, stability, tolerance. We've got to promote trafficking of the antigen to lymph nodes and to get an appropriate type of immune response. So how can we overcome these barriers for these intranasal vaccines? Well, we need to use a proper adjuvant system. This helps us to recognize the antigen as a foreign entity that deserves some attention. Using a proper adjuvant can also reduce the amount of antigen which is being used. That could reduce the cost. Okay, so this is a very important consideration. Also, when we use an adjuvant system, it's not only just an inactive carrier and inactive excipient, it can actually help to modulate the immune response. So we're trying to get a balanced kind of immune response, and our adjuvants can help us to do that. We want to promote mucoadhesion, and that we want to improve residence time in the nasal cavity and not just have really rapid clearance. And we think that is important because it may increase the chance of cellular uptake by those antigen-presenting cells and reduce clearance. So a carrier system needs to promote stability in the nasal environment. And it certainly has to be reproducible in the way we usually think about nanoparticles, usual characterizations, and to have that appropriate response. And hopefully it is a relatively broad enough type of approach such that we can use different kinds of antigens in our carrier system. All right, so let's talk about adjuvants now. I'm working with scientists at Vito Intervac to look at a triple adjuvant, which we're calling triage. And they have been working on variations of this over more than a dozen years, different variations on these combination kinds of novel adjuvants. The current version is based on 3 components. There's poly-IC, which is double-stranded RNA, and this is a toll-like receptor type 3 agonist. So this helps to dendritic cells to become activated, to mature into really phagocytic cell type to get them moving. It also incorporates an innate defense regulator peptide, which modulates the inflammatory response and attracts immune cells. And this class of molecules is under investigation in the laboratory of Dr. Bob Hancock at UBC. Polyphosphazine is kind of an anionic carrier. It's a big polymer which kind of wraps the whole thing up, but it's also a pathogen-associated molecular pattern agonist, so it kind of has its own activity. So over the course of many years and many in vivo studies through scientists at Vito Intervac and their many collaborators, they found that this works really well, used subcutaneously or intramuscularly. And through a lot of in vivo studies, they found that a particular ratio of components was very effective. Now, I come along the project. My job is to try to further enhance this triple adjuvant, to enable it, if you will, for making it work better by the intranasal route of administration to make it into a better mucosal vaccine. So when I first got into this project just a few years ago, and I am pretty new to this vaccine field, but when they told me about this triple adjuvant, the first thing I wanted to do was to kind of investigate this triple adjuvant. And I found that at this ratio, which is very effective that they're using, these macromolecules self-assemble into a kind of a nanoparticle on their own. So it is a submicron kind of self-assembled entity. All right, so what are we trying to do to make this work better? We want it to be relatively mucoadhesive, so it's got to kind of stick there. It's got to bind somewhat to the mucosal surface. Cationic particles do attract recognition by immune cells. We think it needs to be relatively small, maybe to start penetrating the mucus layer. And we set a target of being less than 250 nanometers to be hopefully a good size for dendritic cell phagocytosis, so not really much bigger than that. Okay, so the triple adjuvant triage happens to be anionic because of the ratio of components, so it should bind cationic lipids fairly easily. We know that cationic lipids have been shown to be immunogenic. There are other nasal vaccines in development using cationic lipids or liposomes. And that has been used. And we have the potential, because these cationic lipids have some of their own adjuvant activity, to enhance or modulate the immune response. And what we're trying to do here is to really increase that mucosal IgA level, okay, and IgG2A, which I'll mention a little bit later. All right, so the triple adjuvant is a 1:2:1 weight to weight to weight ratio, poly-IC, double-stranded RNA, this innate defense regulator peptide, and the polyphosphazine polymer. When they're combined, you come up with the triage particles. So this is in an aqueous environment. So we've got nucleic acid and water-soluble polymers and so on. If we want to combine it with cationic lipid, it's got to be able to do that in an aqueous suspension. So what I thought of doing was to have it interact with cationic liposomes. So I come from a liposome background, very familiar with that technology for drug and nucleic acid delivery, so it made sense to me to go about it that way. So using kind of a classic cationic lipid formulation approach, where we can vary the cationic lipid, utilize DOPE as a fusogenic helper lipid, because this material has to escape from endosomes eventually, right? and perhaps some other components to modulate the structure. So in the initial approach, we make liposomes by the thin film extrusion method, and then the liposomes and the triple adjuvant are combined at a particular ratio that allows for self-assembly. I won't get into it today, but I spent a lot of time kind of titrating that proper ratio of components to actually generate a kind of a nanocomplex or nanoparticle kind of combination, which would not aggregate and which would have good activity. Okay, that's another story. All right, so then we come to, after some preliminary formulation work, we have to do some in vivo assessment of immunogenicity. So some things that we would consider in a typical study in this project would be the lipid composition, varying the cationic lipid or the other components and seeing what the effect is. We've looked at low and high doses of the triple adjuvant, so 1 to 2 to 1 or 5 to 10 to 5 weight ratio of the components of the triple adjuvant. Initially, we started working with kind of a test antigen, ovalbumin. So you don't get a lot of response to ovalbumin unless there's a really good adjuvant present. So it's like a screening for a good adjuvant. So ovalbumin is that model. And then later I'll talk about the pertussis antigens. We can look at different doses of the antigen because we want to have that antigen sparing effect, remember. There's a prior formulation using a calcium microparticle. So comparing to previous formulations or just triage alone, and of course the dosing route. Okay. So all in vivo studies, I want to emphasize, all the in vivo studies, all the immunological assays and so on, those are conducted at the Vaccine and Infectious Disease Organization in the laboratory of Dr. Volker Gertz. So I did not conduct those assays myself. It's been a good collaboration. So this is just an example of experimental overview. We might have 5 or 7 or 8 mice per group, and our groups might include phosphate buffered saline as a control, adjuvant alone, antigen alone, like ovalbumin, the ovalbumin combined with the triple adjuvant, and our lipid compositions. For example, in this example, I've got DDAB DOPE versus DDAB APC DOPE to look at the cationic lipid component. Mice are given 20 microliters total volume intranasally by experienced animal care technicians. And mice are vaccinated on day 0, boosted 4 weeks later. And then we can look at ELISA results, immunoglobulin levels, for example, at 4 weeks or 6 weeks or 8, 10 weeks, whatever the study involved. We've also done ELISpot assays where at the end of the study, the spleens are collected from the mice and the lymphocytes isolated, and they challenge with the antigen ex vivo, and then look at cytokine release to see if there's more of a T helper versus cytotoxic T kind of response, or is it balanced, and so on. In the interest of time, a little bit of complexity around those studies, I won't be presenting those today, but they are very similar in result to the immunoglobulin type of results. So here's just an example. Looking in this study at the results of the ELISA assay for IgG1s. This is associated with promoting a humoral type response. And what we have on the y-axis is the titer. So that's the dilution factor needed to get the ELISA response back down to background. So the IgG1 level. And we've got our different groups shown in the legend on the right. So on the x-axis, going from left to right, we've got PBS giving no response, the ovalbumin not giving much response for most. Then we've got in the up-pointing triangles, triple adjuvant with ovalbumin at a fairly high dose. And then looking at the next 4, we've got DDAB DOPE at low or high doses of the triple adjuvant, and another lipid formulation at low versus high doses of the triple adjuvant. So the clear winner is circled in gold as being the most potent, and that's considered to be a decent response there. So the greater proportion of cation lipid was helpful, and a greater amount of the triple adjuvant. Looking at IgG2A response, this is important for developing a cytotoxic cellular response that can attack intracellular pathogens. So this would be really important, for example, for viral pathogens. The IgG2A response, for whatever reason, is a little bit harder to generate from the nasal route of administration, from what I understand. And again, we get our clear winner with the more cation lipid and the higher dose of the L-triage. This is also important to note that this is a 6-week response, and it's a fairly high titer of IgG2A. And so this is an important finding. Getting that really early response to vaccination is important to achieve. Okay, now in a separate study, which is designed in a very similar way, we also looked at a dose response of the antigen, so the albumin, either at 1 or 10 micrograms. And we were comparing to a calcium microparticle formulation, which had been, the method had been previously published. So I didn't come up with that one. That was from another group. And we compared that with the microparticle formulation with our L-triage formulation given to mice intranasally. And this is the week 10 IgG1 response, just as an example. And what we have is that there is a dose response. So we get a greater response with 10 micrograms of albumin compared to 1. Except for the lipid formulations. Here we get a really good response even with a lower level of antigen, and it compares very favorably relative to the microparticle formulation by the intranasal route of administration. So that was very encouraging there. Okay, but what about mucosal immunity? So in that same study that I was just illustrating, at week 10, they also did an intranasal wash. So basically just, flush out the nasal cavity and then looking at the IgA levels, that's the mucosal immunity that we're looking for. And what we found here is that when the albumin was set to, the antigen level was set 10 micrograms, and we used the higher dose of L-triage, we found that the L-triage gave us a really nice level of IgA in the intranasal wash, far more than the calcium microparticle formulation, and we didn't see levels of that when any of those doses were given intramuscularly. So here we have some evidence that we are generating some mucosal immunity through this approach. Alrighty, so we did, number of studies using that test antigen. So then it was time to move forward with more therapeutic vaccines using our actual antigens of interest. And then it kind of begs the question, where exactly should we put these antigens? Do we need to think about that? So kind of a classic approach, which I'm calling LTA for L-triage plus the antigen. We first make the, as I described earlier, we combine cationic liposomes and the triple adjuvant in an aqueous suspension to create a new kind of condensed lipid nanoparticle that we call L-triage. And then the vaccine antigens are simply mixed with the antigen just prior to administration. Okay, so this is a common approach. For example, Shingrix is done this way. There are other examples of that. So we think this is probably going to be the best approach if the antigen has a negative charge, so it can then kind of adsorb on top of the L-triage surface, or maybe it just needs to be nearby, such that we activate the dendritic cells to be somewhat activated, more phagocytic, and take up more of the antigen. I'm not too sure there. But at any rate, the antigen is surface available, and that's one route to go. The other is to incorporate it into the interior of the triple adjuvant during the triage preparation. And so this method has been used before. My collaborators, Dr. Sylvia Vandenherk, published a study using this approach and got some good results. So in that case, the triple adjuvant would contain the antigen during its preparation, and then the cationic carrier is added afterwards, in which case it is fully encapsulated. And we've had good results getting the antigen inside in that case. It's just a schematic where we've got our two different approaches, antigen either on the interior or kind of absorbed on the outside. Okay, so this is important to think about for our pertussis vaccines, because there's actually 3 antigens involved in this. There's pertactin, there's inactivated pertussis toxin, and there's fimbriae, and they're all kind of different, and it's a lot of antigen to pack into a nanoparticle, but we did our best. So we have our triple adjuvant, the triage, and our cationic lipids, and we either packed them on the interior or associated them with the exterior by a simple mixing approach, and then vaccinated our mice and looked for systemic and mucosal immunity. So a lot of this work was done by my postdoc, Dr. Nora Bonney. Okay, so some things we might look at are the effective lipid composition, the lipid to triage ratio. We've done some studies on that, as well as the way in which we incorporate the antigen. Typical characterization would be particle size. Use dynamic light scattering for that, and we're aiming for that, you know, preferably not more than 200 nanometers, thinking that might be an optimal size. Zeta potential, we're looking for cationic nanoparticles, so they won't aggregate too much, and they will be attractive for immune cell phagocytosis. We use transmission EM and scanning EM for looking at the morphology to try to get to pair that with the dynamic light scattering to get an assessment of morphology and size and so on. Entrapment efficiency, mucoadhesion. So that we've looked at zeta potential changes in the presence of mucin, for example, and techniques like surface plasmon resonance. We look at in vitro uptake, cytotoxicity, using fluorescent labels, for example, of various components, and standard assays for looking at tolerability to macrophages or dendritic cells, for example. Okay, so just as an example, this figure is taken from Dr. Nora Bonney's paper, which is currently in review for looking at some of the characterization of our initial prototypes for pertussis vaccines. Again, we've got those 3 antigens in there, and we are building them into our carriers through the 2 different methods. So we've got 2 axes on this graph. On the left side, we've got particle size, and on the right side in green, and the green bars, is theta potential. So on the left, liposomes alone, one of our starting materials that we prepare by extrusion, at 100 nanometer, 120 nanometers, and positive zeta potential. And the triple adjuvant is a lot larger, more polydispersed, and it has a negative zeta potential. The L triage is not much bigger than the liposomes, and is smaller than the triage alone. So it kind of condenses down, and we have a positive zeta potential on that. This is at the ratio that was titrated to effect in the beginning. When we add the antigens to the triage, of course, it's, you know, big, large aggregate sort of thing. And then our lipid formulations, they're in the order of about 200 to 250 nanometers and have a highly positive zeta potential. So these transmission EM micrographs are shown on the right-hand side. LAT at the top, where the antigen is packed in the interior. What we see there is that we've got a kind of a dense sort of a center, and then we see these layers of lipid which kind of go around the outside, and we tend to see multiple layers of lipid. So these are not liposomes. We're not somehow extruding them into become liposomes. It's not like that at all. It's there's layers of lipid on the exterior as they've reformed around those particles through those electrostatic interactions. And when the antigen is added to the exterior of the L triage, we still maintain our particle size in a similar kind of range. Okay. So we're just getting ready to hopefully publish our paper describing all of our in vivo results, but I'll cut to the chase and look at the some key results here. We did find these pertussis vaccines generated good levels of IgG1 and IgG2A serum levels. Looking at the nasal wash IgA, which is demonstrating the mucosal response, we found that for some of the antigens we got a decent IgA level, but it wasn't the same for all the antigens. So Fimbriae, for example, didn't give a very strong response. We got a stronger response for pertactin and pertussis toxin. What we have on this graph here is a normalized dose response. So we set as a level of 1, the 1 microgram of antigen plus triage dose that was given to the mice. We set that as 1, and then everything else is a fold increase above that level. So the pink bars, we have the antigen plus triage alone was given to the mice, and we see 1 versus 5 for the different antigens. And so we have a dose response there as expected. And then for the L triage plus antigen on the exterior, it's shown in the green bars, 1 versus 5. So we get increase in response according to dose, especially for pertussis toxin. And then the LAT, where we have a more monodispersed kind of particles, and the antigens on the interior, we get a dose response again, but perhaps not as much of a response as the L triage plus antigen. Okay, so we have some hint that maybe we can, with this sort of approach, we might get some improvement in our intranasal IgA activity. Okay, we did not have an improvement when the antigen was packed in the interior of the particles. So that's a work in progress. I mentioned particles, so we want to create, really well-defined uniform particles, a scalable approach, and so on. Something that we can use for pretty much any antigens. So we're starting to use microfluidics to make our particles. This is the system which is sitting in my lab. So we've got 3 pumps that can have 3 different solutions or suspensions in them, which pump into the microfluidic chip, which is shown on the right. I've got different chips with different geometries, and you can have any sort of pathway coming in because you can control which lines are coming into the chip and what they contain. Okay. So my former postdoc, Dr. Parth Patel, devised this figure and spent a lot of time working out a method of creating the particles through the microfluidics approach. And this is one scheme that he came up with. So, for example, running off of pump 1, you could have the innate defense regulator peptide and the antigen. Coming off the second pump, the poly-IC and polyphosphazine. Those are both negatively charged, so they won't clump up together. And when they run together, they form the triage complex in line. And then the lipids can be coming off of a third pump, and then in that droplet junction chip, they all come together. If you do it at the right flow rate ratio and so on, and control the temperature, then you can get the LTA or LAT, excuse me, particles. Okay. So you can also work this to have the liposomes can also be prepared on the microfluidics. We're capable of doing that and having that come in line to create the L triage plus antigen also on the microfluidics device. Okay, so similar to the other study, this is now using an antigen for bovine viral diarrhea virus called E2, and that's in collaboration with Dr. Sylvia Vandenherk at UDO. And what we found here with our size and zeta potential characterization is that through the microfluidics approach, we can get well-defined particles that 200 to 250 nanometer range with a positive zeta potential and a low polydispersity index. So that was very encouraging that we were able to do that. We met our goals there. And we just have some fun pictures here. We've got that Parth put together where we've got on the left-hand side, we've got our antigen triage complex and then some liposomes which are in the process of associating with that. Shown on the right-hand side, we've got what we typically see are these layers of lipids surrounding the triage complex. And we can also get, as shown on the bottom left, an aggregate of the lipid and the antigen associated with that. There does seem to be kind of a limit size with that, as the surface of the triage kind of becomes occupied with liposomes. It's not really going to grow any further than that. Using microfluidics, what we see are these more discrete particles which are coated with layers of lipid. They seem to be a little bit more solid, if you will, but they're just covered in layers of that lipid. Okay. And the LAT vaccine particle, where the antigen is on the interior, kind of similarly, we see those layers of lipid around that center. So bringing it to a close, those latter formulations, we are in the process of testing those in vivo. I haven't published those results just yet, but at any rate, the cationic lipid nanoparticles incorporating triage are adjuvants that are generating that balanced immune response. We're getting some indication of a mucosal response, so that's very encouraging. It does depend a bit on the antigen features, and the lipid composition definitely has an influence, as well as the fabrication method. It does seem to work a little bit better when the antigen is associated with the exterior as opposed to buried on the interior. And we are able to make these either by simple admixture procedure or by the microfluidics approach. So we've met our goal of creating particles in the size that we set out. Okay, challenges, limitations. Okay. Like most science, it raises many questions as we go. Definitely, we have ongoing challenges as we continue to develop our various vaccines. So pertussis is really tough because there are 30 antigens, and it's so much material that we are incorporating into these vaccines. They're a little bit different from each other, so that's an ongoing challenge. We do have kind of a limited set of lipids that we can work with. We have to be able to purchase it. So we don't have endless libraries, so we have to kind of choose candidates that are different enough that we can try to get a sense of what the important features are. Those particle properties are really important. So we want to be studying what is the optimal organization of those components. I'm not convinced we know what the optimal size is just yet for getting that response. But we want to have good control on the morphology of our particles for sure. We're moving forward to looking at the intracellular processing of our particles in our antigen-presenting cells. Those antigens need to be able to escape from the carrier and to actually be presented in the context of MHC1 and 2 in order to be effective. So we need to look at that closely, and we need to work on building some mucosal epithelial models in the near future. So moving forward into future vaccine studies in vivo, however, we do look forward to working with Dr. Gertz in his neonatal pig model of pertussis challenge that he's developed. We're going to be looking at, I didn't talk about influenza today, just there's only so much time, but we'll be doing some H1N1 challenge studies through NMIN's PharmaCore. They have an influenza model there. And then with Dr. Vandenherk, when we have good mucosal response with our various candidates, we have the opportunity to do a large animal study with her group as well. Okay, and then I just want to acknowledge my fantastic collaborators who've done so much work on this project. Dr. Gertz's lab and his technicians, and Dr. Vandenherk, Jens is one of our collaborators on the influenza aspect, and they're co-investigators at UBC as well. A lot of the slides I presented today have data generated by Nora Bani and Parth Patel. Nora did so much work on the Pertussis project. And I have these new students coming in who just started in the fall, and they'll be continuing on in the project and its many challenges. I acknowledge funding from the Government of Saskatchewan, the Agricultural Development Branch, Saskatchewan Cattlemen's Association, certainly NMIN. Gave a lot of in-kind support early on, and they're my partner in many grants, and CIHR for their funding as well. And I thank you very much for your time today and coming out to listen to this new project. Thank you very much.
Well, that was an excellent seminar, Ellen, very comprehensive and impressive studies. Thank you. Before we began the video, I mentioned that it is International Day of Women and Girls in Science. And also then for this reason, just how great it is to have you as our featured speaker. I also noted during the seminar, just seeing some of the names here of people that I know in the participant list, that we have folks present from all over the world, which is just amazing to see. So clearly people are very interested in seeing you and listening to your seminar. So we've got first from Francisco Passini. How do you sterilize the lipid particles for the tests? Do you use a 0.22 micron filter?
Okay, that is a great question. That is a great question. We are always concerned about such matters. We take effort to, clean our supplies, autoclave things, use, sterile diluents and things of that nature, open the biological safety cabinet wherever possible. Because of the nature of the particles, it's not possible to do final sterile filtration. They're just a little bit too big for a 0.2 micron filter at this point. So the way that we have dealt with that is just we look for a specific immune response, and we're not creating an infection with our materials, but it's something we'll have to look on in the future, how we would do terminal sterilization and things like that. This is research based in an academic lab. So at this point, all we can do is try to be as clean as possible, and most of our materials are filtered in some way prior to use.
Perfect. Perfect. Thanks, Ellen. Our next question is about sterilization of the microfluidic device. I wanted to just also ask you, I don't know if it's a student or postdoc that put together those beautiful schematics of the, really quite impressive of the formula.
I know, he's good at that.
Yeah. It's like, wow. I just had texted to some of the hosts and panelists here that this is very cool research. I just wanted, we're starting to get into use of microfluidic devices for formulation prep. And, do you see real advantages? Do you think this is the future of kind of nano formulation prep, or is it a lot of hype and just a new technology that people are excited about right now?
I think it's a bit of both.
Okay. Okay.
We both know that everyone is very excited about microfluidics right now, especially in light of the current era of vaccines. And absolutely, it has a lot of functionality. I've asked people in industry, what do they think about the actual, process of scale up? Is it as simple as it seems to be? Yeah. And I would leave those kind of questions to them that are dealing with more of the scale up issues. Obviously, they've figured out ways to do that. Yeah. I've noticed differences in activity between the particles that we make with the simple batch mixing or ad mixing kind of procedures versus the microfluidics prepared ones. And so I have to investigate that a little bit different. What is actually different in the structure? They are different. They are assembled differently, and that affects the way they're going to be handled by cells. And that's something that we have to look at more directly. So it's not a direct translation of the more traditional techniques straight to microfluidics. You have to, I think, re-optimize again, and for activity, for stability. It's not a direct one-to-one. And I guess you do save on time for certain steps. Yeah. When we make liposomes, it takes a couple days. Just in yes. Traditional extrusion methods, and you can do it quickly on microfluidics, but then you have to get rid of your ethanol, and that's another step. So and it's as far as sterilizing the microfluidic device, you can easily run ethanol through there. Okay. Great. Yeah.
Great. Thank you. The next question from Lorenzo: would you see your particles working also for mRNA type vaccines?
Sure, why not?
Sure, why not? Perfect. Great answer.
I mean, if you need to take a look at the way that the current COVID vaccines are constructed, and I encourage you to read the literature around the development of those vaccines, the research papers, and you'll see how they are constructed. And absolutely, a similar approach is being used there. And that came out of decades of research that was going on. And I started this project before all of that really came to the forefront. It's just all kind of gelling together now.
That's amazing.
Absolutely.
Yes.
Yeah.
Very hot field. That's great. We've got quite a few people here saying that it was a great talk. It seems this is the future of vaccine technology. Another person saying, thanks for the excellent webinar. Great presentation. Thank you. So another question from Yime Jia. My question, any reason to use poly-IC? Have you tried MF59 adjuvant, which is approved to use in the influenza vaccine?
I have not tried MF59 specifically. This project came about as finding a way to enable this triple adjuvant. Triage was my API, basically.
Right,
I was formulating the triple adjuvant, which had already been developed through many studies, which I try to emphasize in this seminar. They looked at CpG, poly-IC, they looked at different peptides, different ratios of components. And they did a lot of the optimization in vivo. So they would look at these different combinations, and maybe it wasn't a situation where they were characterizing a lot of it in a pharmaceutical way that we would think about doing it out of this group, but they did a lot of the important work in vivo where it counts. So that's where the triple adjuvant comes from.
Okay.
So could MF59 be a good choice in this case? Maybe so, but that would have been a little bit outside of the scope of what my goals were for this project, which was to develop a nasal formulation, try to get some additional immune response for the mucosal response through the nasal route. So yeah.
Thank you. We've got another couple of people saying excellent talk. So I think we've come to the end of our questions that are here.
Okay.
Maybe I'd just ask you, you started at the beginning. I loved how you kind of identified all the criteria that you were aiming for this formulation. And you showed all this beautiful work, and just give people a sense, maybe, because I know we've got a lot of trainees on here, just how long this research took, because it all comes together as this beautiful story. Can you give us a sense? Like it's quite a nice, tight story, but is this years of work or?
Not that many. I still consider this to be a fairly new project. And
wow.
I am new to immunology, new to vaccine work. I've been in drug delivery a long time. I did some work with gene therapy and things like that. So I'm new to the whole immunology aspect, and I leave a lot of that part to my collaborators across campus. But I would say I started working on it in 2016, and the first thing to do was to figure out what is this triple adjuvant. So we spent a lot of time figuring out what is this thing, how can we characterize it, how can we do quantitative assays, try to understand it better. So that took some time, and then try to figure out how can we associate it with our cationic lipids, and then that took another year. Just figuring out how to make it not crash, trying to figure out the right kind of ratios to make it into a proper articulate sort of lipid delivery system. So all the characterization around that probably took another year. And then we have done, we did initially 3 in vivo studies with the old albumin as the model antigen, which is this gold standard for looking at adjuvant activity. And, those studies took another year and a half, 2 years, so 2018. And then we published around that. And then we started looking at our therapeutic vaccines. So we did, we've done 3 studies with the bovine viral diarrhea virus, which is aimed at cattle shipping fever. And we've done 1 study in vivo with pertussis. And then the pertussis development is, I would say, still in process. And we probably spent, Nora's whole postdoc on that. So a couple of years, she spent working on that aspect. Yeah. So I would say since like 2016 or so.
Wow. Oh, that's fantastic.
And it's still early days. It still feels like early days. Like we have so much to do to understand how to, to work this better with, the resources that we have.
No, it's exciting. A couple more questions. One is the microfluidic device commercially available or is it homemade?
No, it's commercially available. It was from Dolomite Microfluidics, which is mostly out of the UK, but they have offices throughout the world. So it's a Dolomite system that they call a nanoparticle generator. But basically, it's just like I showed you, it's a series of pumps. You can get a bunch of pumps, you can get a bunch of chips, and you can have different geometries of chips that are commercially available. So I don't make the chips. I don't work with anybody to make the chips. You can purchase them for $600, $700, $800. So yeah, we have to be careful with those.
Right.
They are,
So we do have one more question. What are the possibilities that drug formulation in liposomes, Doxil, for example, can be done in lipid nanoparticles? Do you foresee any obstacles? So I think this person may be asking if you see this as a strategy for small molecules like doxorubicin.
Well, I think liposomal doxorubicin is working extremely well.
Yeah,
in its commercial form. So I don't think I want to mess with that. It works. It's a commercial product. It works great. You know, why change it? So you'd have to have a reason. You know, you can. We can make liposomes on our microfluidic device.
Yes.
Quite readily. You just change the parameters. So certainly, it can be used for that.
Well, I'm delighted to hear that you got the Dolomite system because this is exactly what we're in the midst of purchasing. And I thought we might be the only ones going with Dolomite versus some of the other vendors out there. So I'm really gaining some peace of mind here, hearing that it's working for you. So that's great. And you've got such fantastic results. So that's wonderful. I can see Diana has her hand up.
Hi, Ellen. How are you?
Good, thanks. How are you?
Thanks. I'm terrific. Thank you so much for a wonderful presentation today. And I just want to say how illustrative of the power of networking your talk was, and what exciting research happens when you're working with people who have different expertise than you do. And so when you talked about your collaborators and learning about all these new techniques and new ways to apply your expertise, I really appreciate that you've shared this with everyone today, because it truly is a network way of working, and I want to congratulate you on that regard.
Thank you, NMIN.
Great. Thank you. So thank you so much, Ellen, for delivering the February NMIN lecture. I have to say it's pretty special for me to see you here today. It's like I said before, International Day of Girls and Women in Science. You're an outstanding scientist, a great role model for so many women. And to our participants, thank you for joining us today.